Hey everybody, today we'll be discussing the immune system, and any discussion of the immune system has to begin with the different layers of defense that comprise the immune system. So the first line of defense that you may not think of as being part of an immune function, but it certainly is, are the cutaneous and mucous membranes that make up your skin, and also the epithelium that secretes mucus and other compounds that can have the effect of providing immunity. So skin being a barrier between the internal and external environments, of course that's going to contribute to immunity. And skin containing this protein keratin is going to be key to it being able to provide a waterproof barrier that keeps whatever's inside separate from whatever's outside. So keratin is an important part of why our skin is able to provide such an effective barrier. Also in those cutaneous membranes, you have oil glands, or the sebaceous glands that secrete sebum, and that has antimicrobial properties as well, and is partly due to the fact that it's got an acidic pH. So here I mentioned acid as well, so not only acid in the sebum, but in the mucosa there are also, particularly in the stomach, there are going to be cells that secrete acid that can deter the growth of harmful microbes. Your certain portions of your skin also secrete molecules called defensins, which have antimicrobial properties. And the normal flora, you may have heard, a lot of recent advancements have come to define really important properties of the bacteria that normally are a part of your body, and how they can help to fight off foreign invaders and fight off infections. So you have a normal flora. That term flora just describes the bacteria that kind of live in a symbiotic relationship with us, and they help us to fight off what would be an invasive or infectious microorganisms. And viruses for that matter as well. So the first line of defense are the membranes that are on the outside of your body, and also lining the mucosa. The other two, which are somewhat related in how they respond, but you have the innate immune system and the adaptive immune system. So innate just means that this arm of the immune system requires no previous experience with a pathogen in order to have an effect. It's non-specific. So your innate immune system is going to fight off any sort of invasive organisms, whereas the adaptive immune system has a specific response. And it's mediated by cells that can recognize specific antigens or pathogens. So we'll get into more about what an antigen is later, but an antigen is what's going to be part of any invader that's going to allow it to be recognized by the adaptive immune system. So innate is kind of your inborn immune system that'll fight off any substance or any pathogen that enters your body in a non-specific way. But again, the adaptive immune system is going to require a memory of a certain pathogen. So the adaptive immune system you can think about being the reason why we get vaccines, which is especially relevant now with the pandemic, is that once you've been introduced to a particular pathogen, your immune system learns to recognize that pathogen so that it can mount an effective response the next time it sees it. So that's what the adaptive immune system is all about. the innate immune system is going to provide a rapid response to any pathogenic challenge. So a pathogen is introduced into the body, and this quick response within the first 12 hours of exposure to a pathogen, the cells and proteins of the innate immune system are already present in the blood. So in order to mount a defense via the innate immune system, it's very rapid because, again, those cells and proteins involved in the innate immune system response are already there waiting to rise to the challenge. So while the adaptive immune system takes a little bit longer than the innate immune system to mount that response, it can be incredibly powerful, and that's based on using antibodies to generate specificity. So what that means is the adaptive immune system is able to respond to the presence of an antigen. Now, what an antigen is any little physical part of a pathogen, for instance, the coronavirus. It's a specific part of a pathogen that the immune system will remember, will have a memory, biological memory of. And that allows it to target the immune system to target it much more efficiently. So let's take the coronavirus, for example. You may have seen pictures of what the virus particle looks like, and it's got this little spherical capsid, the outer layer around the virus, with these little spike proteins on it. And those spikes apparently are what allow the virus to recognize our cells and to transport themselves inside our cells. So these spike proteins are thought to be a great candidate for an antigen, or a part of the virus that we could train our immune system to recognize. So that's the basis of a vaccine, would be introducing your body to just the spike proteins, for example. So one vaccine candidate may involve injecting your patients with just the spike proteins, not the entire virus, but just these spikes, and hoping that our immune systems mount an immune response against just those spike proteins. So this, of course, wouldn't cause an infection because you don't have the virus, the entire virus, you just have the spikes. But if your body was able to mount a response to those spike proteins, then if they encountered the actual virus, you would be able to fight off that infection much more efficiently because your immune system would remember those spikes. So those spikes would be the antigen that's being recognized with this biological memory that makes the adaptive immune system so efficient. So an adaptive immune response is going to take a little bit longer, at least for the first time you encounter a new pathogen. So for your first exposure, it takes about 3 to 5 days to mount an immune response via the adaptive immune system. And this response is going to consist of responses in the two arms of the adaptive immune system. The first arm being the cell-mediated arm, which consists of a response of these cells called T cells. And we've already talked about T cells a little bit. They mature in the thymus. And the other arm of the adaptive immune system is going to be the antibody-mediated arm. And this is regulated by B cells. So B cells are going to be what are actually producing antibodies. And what an antibody is just going to be a protein that your body, that those B cells create, that can recognize an antigen. So these antibodies usually look like little Ys. And antibodies stick to an antigen and allow that pathogen to be recognized by the immune system. So B cells create antibodies that are circulated through the blood, and they will stick to a specific pathogen. So these antibodies that I've drawn here recognize specifically the spike protein antigens found on the coronavirus. I'd like to quickly go over the primary immune cells that are going to be active in either the innate or the adaptive immune system. And primarily these consist of, they're all white blood cells, but we kind of group the white blood cells or the leukocytes into either agranulocytes or granulocytes. And what that means, that term granulocyte, means that if you have a cell like that, if it's got what are called granules, a granule is going to be a little package, a vesicle basically, inside of the cell that contains some substance. And that substance is going to be critical to the function of that cell. So a granulocyte is going to release that substance out of those granules in an immune response. So sometimes those granules can contain inflammatory mediators. So jumping ahead a little bit, these basophils down here are granulocytes that release chemicals that promote inflammation. Other granulocytes, your eosinophils, are going to contain substances that kill parasites. So granulocytes basically have these little speckles that you can see under the microscope if you stain them. And agranulocytes do not have those granules. So that's the difference between the agranulocytes and the granulocytes. The agranulocytes consist of B and T cells, which we've already described. B cells producing antibodies, and T cells are involved in activating other immune cells. Also, monocytes are going to be a phagocytic. So monocytes are phagocytes. They go around ingesting bacteria and debris. Now, the granulocytes, I've already mentioned the basophils promote inflammation by releasing granules with inflammatory mediators. Eosinophils are going to directly target parasites, such as parasitic worms in particular. And neutrophils are going to be a type of granulocyte that is also a phagocyte. Now, another type of cell that's involved in the immune system is the dendritic cells, which we mentioned before live in some lymphoid organs, and their job is going to be to activate B and T cells. Aside from the cells that are clearly important in carrying out various activities of the immune system, proteins are an important part of the immune response as well. And we've already described the effect of antibodies in generating a immunological memory of certain pathogens. Another important set of proteins involved in the innate immune system, as opposed to antibodies, which are part of the adaptive immune system, is the complement proteins. And these complement proteins are produced by the liver, and they're circulating in the blood waiting to be activated under certain circumstances. Now, the complement proteins can have a variety of effects to help fight off an infection. And we'll get into those effects a little bit more deeply in the mechanism of activating the complement system. But the complement proteins are always circulating in the blood, but a specific event has to occur before they become activated, where they can have this non-specific effect of having an antimicrobial effect. And then the last kind of class of proteins that are involved in the immune responses, both innate and adaptive, are cytokines. And cytokines are basically chemical messengers that are going to be secreted under certain circumstances, and they can have a variety of effects, either by activating certain cells that are part of an immune response, or they can also promote maturation of cells and ultimately that's going to facilitate an immune response. So they can help to mature immune cells, or they can also regulate the activity of those immune cells, whether they're actively seeking and destroying or whether they're in a more passive state. So I'd like to talk about the cells that make up the innate and also eventually the adaptive immune system. When we're talking about the innate immune system, you basically can classify the types of cells by what their function is in generating immunity. And the first class of these cells are going to be the phagocytes. So you probably remember the term phagocytosis, which means cell eating. And during that process, a cell is going to be attracted to whatever it needs to eat. In this case, we're talking about pathogens like bacteria. It's going to be attracted to them, and it's going to move toward them by this process of chemotaxis. So what chemotaxis means is that there's a chemical signature of the bacteria, for example. And these phagocytes can basically sniff out that chemical signature and move toward it. So these cells are going to be chemotactic. And once they approach whatever needs to be swallowed, they're going to wrap these structures around called pseudopods. Once they've done that, they'll pinch off part of their plasma membrane and form an intracellular vesicle that contains whatever it is they swallowed. In this case, again, we're saying this is a pathogen, it's a bacterium. Once that endosome containing the bacteria is inside the cell, it's going to fuse with another vesicle. So what I've drawn here is going to be a lysosome. You might remember that the lysosomes are organelles that contain digestive enzymes. So if a cell is going to swallow something, whatever that is needs to be digested. And the lysosomes contain those enzymes that help it go about that process to destroy this bacteria. So these two vesicles are going to fuse together, and that's going to destroy the bacteria when it's exposed to those digestive enzymes. So sometimes these cells that are phagocytes can also not only destroy a bacterium, but they can also facilitate other parts of the immune response. And what I mean by that is that some of these phagocytes can be what are called antigen-presenting cells, or APCs. So we've already talked about what an antigen is. It's just a piece of the pathogen that can be used to identify that pathogen. And some of these phagocytes, once they've destroyed the pathogen, they're going to attach an antigen, or a part of that pathogen, to their own plasma membrane and present it to other cells of the immune system. Basically, it's like your cat, dragging in a dead animal and presenting it to you, saying, "Here, this is what I've got for you." It's as if they're saying, there's a rat invasion. Look at this. So these cells are going to do that. They're going to present these antigens to other cells that activate them. So an antigen-presenting cell can bring in other phagocytes, and they'll be activated by contacting the cell that's presenting the antigen. They can also activate things like B cells and T cells that can be, part of, B cells generating antibodies against that antigen, for example. So APCs are going to help to activate other parts of the immune system, and that's useful as well as just destroying the pathogen. So these phagocytes include macrophages and neutrophils, which are pretty simple in how they get rid of pathogens. They just swallow them and digest them. Eosinophils, again, are those, they're phagocytes, but they also are going to specifically target parasites like parasitic worms. And then you have dendritic cells, which are phagocytes, and they're sometimes going to be antigen-presenting cells. The other division of cells in the innate immune system are going to be your non-phagocytes. And natural killer cells make up one type of cell that's not phagocytic, but it is cytotoxic, meaning it can release toxins that are going to help to kill pathogens. It just doesn't swallow them. So these natural killer cells are going to be constantly looking around for irregular cells. And by irregular, I mean these are cells that are out of the ordinary or not normal body cells. So these can be cells that are bound by antibodies. So antibodies, again, target antigens. They're created by your adaptive immune system. But these natural killer cells of the innate immune system can seek out and destroy cells that are bound by antibodies. So that's one thing that I want to mention is that the innate immune system and the adaptive immune system can cooperate between each other. There is some cooperation. They're not completely distinct from one another. So that can be a little confusing, but these natural killer cells, again, they can cooperate and seek and destroy antibody-bound cells. And these are going to be, again, pathogens that your body's mounted an adaptive immune response against. But these NK cells that are a part of the innate immune system can help to destroy them. Also, another definition of irregular cell is not just one that's bound by antibodies, but your immune system can kill your own cells of your body in some cases. And a case where this would occur would be if a cell is cancerous. So one of the hallmarks of metastatic cancers is that they are able to evade the immune system. Those really dangerous cancers can evade your immune system. But a lot of the time, a cancer cell can form and be cleared by the immune system. And natural killer cells play a big part in this. So NK cells are going to be lymphocytes. They're found in the lymphatic system. They kind of migrate through the lymphatic system. And they're going to again, seek out and destroy these abnormal cells, including cancer cells and also cells bound by antibodies. The other two non-phagocytes are going to be similar in their mechanism, and these are basophils and mast cells. So they're closely related to one another, and they both mediate inflammation. So their response, their part of the immune response is to promote inflammation in the case of something like an infection. The difference between these two is that basophils are going to mediate inflammation in the blood, and mast cells mediate inflammation in mucous membranes. So someone who has bad allergies, their mast cells are going to be triggered by the presence of some allergen, and that's why you get inflammation in like your respiratory tract is due to the abnormal activity of mast cells. But in their they're also very useful. Those are places you don't want to see infections in, and inflammation can help to prevent prevent the spread of infection. So mast cells and basophils are very similar, but they differ in the location of their activity. In addition to cells that are already present in the body that are ready to go at the beginning of any challenge from a pathogen, you also have proteins that are part of the innate immune system that are always present and waiting on that challenge. The first of these is going to be called the complement system of proteins. So this complement system consists of about 30 proteins that are manufactured by the liver. And in their inactive form, when there's no pathogen present, you're going to see these inactive complement proteins just waiting to be activated. So when there's a pathogen present, there's three different mechanisms And these are going to be the classical, the alternative, and the lectin pathways, which I'll encourage you to look at your book to see more about those different pathways. But what these three different pathways are is three different ways that this complement protein, and complement proteins are named C and then a number. So this complement protein C3 is normally floating around in its inactive form. But in the presence of a pathogen, through one of these three pathways, C3 can be activated. And part of that activation is actually chopping this protein or cleaving it into its active form called C3b. So once you've activated C3b, C3b itself, this immune protein, this complement protein, can surround viruses and neutralize them. It can also increase the rate of phagocytosis, so it can help phagocytes to ingest pathogens. And this is through a process called opsonization. And it can also help to clear immune complexes. So active C3b, when it comes in contact with antibodies that are bound to antigens, is going to increase the rate at which they're cleared. So getting rid of things that antibodies are bound to is part of the immune response, and the C3b complement protein helps with that. Another thing that C3b does is it can cause activation of another complement protein called C5. Now C5 is pretty cool. So let's say this is a bacteria, and C5 has been activated. C5 is going to form what's called the membrane attack complex, or the MAC. And what this membrane attack complex is, it's going to look like kind of like a needle or a rod, this hollow tube that gets injected into the membranes of bacteria. So what it's going to do is basically poke holes in a bacterial outer membrane and cause cell lysis. So that's going to cause, this cell to break open and effectively kill a pathogen. So this membrane attack complex is a really handy tool to kill bacteria. And also active C5 is going to kind of encourage basophils and mast cells to increase their production of inflammatory mediators. So when a basophil or a mast cell comes in contact with active C5, it's going to become activated and start to increase inflammation around that area. Another class of antimicrobial proteins are the cytokines. And the first of these is called tumor necrosis factor, which has been named for the fact that it was discovered because of its capacity to kill tumors or cancer cells. So its effect is going to be to encourage flu-like symptoms. Oftentimes when you think of having an infection, you think of getting chills, fever, and having those be an effect perhaps of the pathogen. But most of those flu-like symptoms are actually brought about by your own immune system. In particular, chemical messengers like cytokines can promote those sorts of symptoms. When macrophages are activated, they're going to secrete TNF. And this secretion, TNF can attract other phagocytes. So, and also to increase their activity. And phagocytes can also release more cytokines, and TNF is going to cause that increase in cytokine production. So a lot of the way the immune system works is actually going to be through positive feedback loops. So that's definitely important to remember. Kind of like childbirth has this positive feedback loop that occurs where the cervix stretches and you get pushing of the baby until finally the baby's born. Well, here you get an increase in the number of phagocytes and the number of cytokine that are released, and also an increase in activity. And this feeds back into the system and increases until the challenge has been cleared. So positive feedback loops are good in this case to help destroy pathogens as efficiently as possible. But occasionally an overactive immune system can have some negative effects. So overactive TNF can sometimes lead to septic shock and decrease in blood pressure, blood glucose, that could actually be quite bad in some cases. So an overactive immune system, not good, but tumor necrosis factor in general can have these good effects of attracting phagocytes and causing more cytokines to be released. The other cytokines are going to be the interferons, which are appropriately named because they interfere with pathogenic infections. Specifically, they're going to inhibit the ability of a virus to replicate. So viruses are going to depend on the host cell. So they invade a host cell and they hijack the cell's machinery for making things like proteins and synthesizing DNA and RNA. So a virus cannot replicate without the machinery of the cell itself. It has to hijack those systems in order for it to reproduce. And interferons, which are produced by a variety of white blood cells, are going to interfere with that process. One way that they can do this is to slow down protein production of cells that are bound to interferons. So if the cell itself stops making proteins during a viral infection, then that means it's going to stop making the proteins that the virus needs to replicate itself. And so that can slow down an infection, and that can be very useful. It's also going to have the effect on these cells of increasing their production, in some cases, of antimicrobial proteins. The last type of cytokine that we'll discuss are the interleukins. And they're named interleukins because they're passed between leukocytes. So the white blood cells produce these, and they have effects on other white blood cells, including increasing the rate of neutrophil production. So neutrophils are those phagocytic white blood cells. And that production is going to occur in the bone marrow. So you increase the numbers of neutrophils by producing these interleukins. And natural killer and T cells can also be stimulated by interleukins. Finally, interleukins are going to increase the production of interferons. So these are all going to be helpful in fighting off an infection. So when I return with part 2 of this lecture, we're going to move on into the specifics of what happens initially during an infection, including the inflammatory response. So what's going to cause inflammation, what's the purpose of inflammation, and things like a fever. So we'll talk about that and also then move on into the adaptive immune system.